My name is Michelle Dapanta. The topic is exome sequencing and the management of the neurometabolic disorder. Definition: exome sequencing is a technique for sequencing all the expressed genes in a genome. Neurometabolic disorders: various disorders result from genetically determined abnormalities of enzymes, the metabolic sequence of which affect development or function of the nervous system. Although most occur in children, some can present in adult life. An increasing number of affected children survive into adult life.
Previously, we learned what whole exome sequencing is. Now, we will introduce the bioinformatics pipeline. Specifically, we will talk about how our team of scientists read the DNA sequences to discover what genetic mutations may be the cause of your child's symptoms. After sequencing your DNA, a file of data is produced that can be as large as the size of a 16-gigabyte iPhone. How do these large files of sequenced DNA from TGen's laboratory bench reach the clinic bedside in finding a diagnosis for our patients? The first step in analyzing the patient's sequence exome is to identify the variants, or changes. We do this by comparing your DNA sequences to the reference sequence, or the original human genome that was sequenced by the Human Genome Project. It is similar to assembling a puzzle of DNA fragments by looking at the completed puzzle picture. On average, there are about 15,000 variants, or changes in the DNA sequence of the exome in any person we sequence. Variants in the DNA sequence can arise in many different ways. One way is through a single nucleotide or base pair change. This is where one base is substituted for another. Another way is through an insertion of random bases that were not present in the original sequence. Sometimes, variations can even arise through deletions. In this case, bases from the original sequence are randomly removed. These variants make our DNA unique to us, but not all variants are indicative of disease. Let's use a common word, for example. Say you have the word old. At some point, another base, G, was added to the sequence, making it read gold. Since this still makes sense, the variation is harmless, and the person would be unaffected. But if the D was removed, the new sequence reads GOL, which has no meaning, therefore would negatively affect the protein and gives disease. Sometimes it is difficult to identify the cause of a disease because the deletion or insertion was so large, sequencing could not identify it. Instead, a chromosomal microarray can unveil some of these large variations. With over 20,000 protein encoding genes, how can a variant in one gene be pinpointed as the cause of a disease? This analysis process is part human and part computer.
And I, along with a team of other scientists, filter through the list of genetic variants and compare the patient's genotype to that of their parents' genotypes. And this allows us to determine if the variant is de novo, recessive, dominant, X-linked, or mitochondrial. We then use several different tools that help us to predict how damaging or pathogenic the variant is, and also use publicly available databases to determine the frequency of the variant in the general population. Lastly, we check published journal articles to see if the gene that the variant is in has been previously associated with disease.
There are some limitations to whole exome sequencing and our knowledge of gene function. At the C4RCD, we have an amazing diagnosis rate of 30 to 40%. However, this means that some families still go undiagnosed.
So one of the reasons is that we do not know the role of all the genes in a cell. We may find a variant in a gene, but because we don't know its function, we cannot connect that variant to the disease. Another reason would be that there are limitations to the technology. Exome sequencing is not very well at capturing repetitive regions or small deletions. A third reason would be is that mutations are not always present in the exome, but they could also be in a region often referred to as junk DNA, although it could still be associated with disease. And last, we don't give up. We're trying new technologies to increase our diagnostic rate at all times.
As research progresses and technology is advanced, we are hoping that genomic medicine can be improved and more diagnoses can be made for many unsolved patient cases with continuous reanalysis. Thank you for watching part 2 of this video series. If you have any questions, please ask one of the members of the C4RCD staff.
Application in medicine. Next generation sequencing aims to reduce the recovery of genes in which variants cause rare medley disease. A diagnostic yield of 16 has been documented for whole exome sequencing among patients with unexplained intellectual development disorder, a condition that affects an estimated 3% of the population worldwide, along with cognitive illness that includes epilepsy, psychiatry or high-level disturbance, movement disorders, sensory deficits, and other organ dysfunctions. Intellectual developmental disorders pose a substantial emotional, functional, and economic burden. Copying number variants, methylation abnormalities, and single gene defects are known to cause intellectual developmental disorder.
The human body is made up of 46 chromosomes that come in 23 pairs, one set from our mother and one set from our father. Chromosomes are like different books in an encyclopedia set. Each book carries different information, yet altogether they make up a very important reference set. Our chromosomes are like these books in the encyclopedia set. If one is added, deleted, or out of place, it may cause disruptions in the reference information. The severity of this disruption may depend on how much information is changed as a whole. If we take a closer look at each volume of the encyclopedia set, we will notice that there are pages upon pages of information in each one of these books. These pages are analogs to the genes that code for everything in our body, from tissues to blood to different bodily functions. If these genes are misspelled, misplaced, or moved to a different location within our genome, it can have mild to severe effects on a person's health. But how exactly does this happen? Just like the words on the pages of the encyclopedia, our genes are made up of 4 letters: A, G, C, and T. These 4 letters create the genetic code. In our written language, we expect the letters in words to be in a certain order for our brains to translate the correct meaning. If a letter is added, out of place, or deleted, the word itself may not be understood. Our genetic code works along a very similar translational system. A gene is made up of letters that need to be in a specific order for healthy proteins to be made. If a letter is out of place, added, or deleted, it too may not be understood by the human body. We call this a genetic alteration or a mutation. Some genetic alterations are considered benign or not harmful. These spelling changes in our genes may even be beneficial as they introduce genetic variation in our species. Benign changes may be the cause of humans having different eye color, different hair color, or even different personalities. However, if a genetic alteration is severe or in a part of a gene that has high functional value, it can be the cause of human disease or death. Many times, we call these harmful spelling mistakes in the gene mutations or pathogenic alterations. We are learning more and more about the causes of these spelling mistakes on a daily basis. It is not always clear why some people carry these gene changes while other people do not. In fact, many gene changes happen sporadically in people with no family history of the same change. This can often be difficult to diagnose and can sometimes be confusing for the patient, as they often wonder why this has happened to them. In the past, clinicians have been able to detect these spelling mistakes by sequencing suspect genes one at a time. This often resulted in a costly and time-consuming process. With the advent of clinical exome sequencing, clinicians are now able to simultaneously survey all 20,000 genes. Clinical exome sequencing allows clinicians to better treat and manage their patient's disease by helping them understand the underlying genetic cause. The human body is a complex system, and there is still so much more to be understood. As researchers and doctors continue to learn more about the human body, we will also learn more about how to create better treatments and cures for genetic conditions.
Pros and cons. Pros: high coverage in target regions, reduced costs for large genomes in people, and cons: mutation irregular regions will be missed. Genetic markers can only be genotyped if they are in the target region. Issues applied in Ecuador.
Hi everyone, I am Dr. Chandrakumar, working as consultant neonatologist in Cloud9 Chennai. I am specialized in the care of newborn babies neonatology. So metabolic disorders are due to deficiency of a particular enzyme which is necessary to convert a metabolite in the body to a different metabolite. That is, you have to break down the food which the baby eats. If a baby is drinking milk, it will have protein, fat and carbohydrate. So each and every energy source has to be broken down and then eliminated by the body. So it has to be used for synthesis purpose or storage purpose, or it has to be broken down and the energy derived from the metabolite is used for bodybuilding or other activities, and the remaining breakdown products are eliminated by the body. So if the body has a deficiency of one of the enzyme which is involved in these multiple steps, this breakdown or synthesis cannot happen in the body, which results in accumulation of abnormal metabolites or deficiency of some essential metabolites. So this excess of one metabolite or deficiency of other metabolite results in the various manifestations, various abnormalities in the baby, which can present as metabolic diseases in the newborn. Usually these metabolic diseases are genetically acquired, so there may be different forms of inheritance. Inheritance in the means that the baby gets these diseases by different mutations. So if the parents can be carrier of such diseases, and this can affect the baby subsequently. So based on the mode of inheritance, the severity with which the baby or the number of babies, whether all the babies would be affected or only few babies will be affected, would different depending on the mode of inheritance of these metabolic diseases. So the types of metabolic diseases can be grouped under three major categories, which is something related to carbohydrate metabolism, protein metabolism and fat metabolism. These three are the major groups. In addition, there are metabolic diseases which affect the other systems also. For example, we have what is called as nucleic acid in our DNA and RNA. So there can be some abnormalities related to this nucleic acids also. So there can be some abnormalities in the RBCs, like where the hemoglobin is there. The heme synthesis also there can be abnormalities, which results in a set of metabolic problem. So metabolic problems are can occur in almost every system in the body, and they have wide variety of presentations. So the causes mainly being the genetic, and it is often inherited from one of the parents, or it can be a de novo new mutation which happens in that person for the first time, and might not have occurred in the family prior to the particular index child.
The conclusion: well exome sequencing has both transformed gene diagnostic and rare disease translocation into disease-modified treatments is challenging particularly from intellectual developmental disorder. However, the section is inborn errors of metabolism. Signs many of these disorders are responsive to therapy that targets modification pathology features at the molecular or cellular level. Thank you.